Welcome to the fourth session of the International IVF Initiative. There are approximately 1,810 people that have registered for this session. I am honored and excited to be co-hosting this session of the International IVF Initiative with Jacques Cohen. For videos of previous sessions, please go to ivfmeeting.com. You will also find information about upcoming sessions on that same page. Our continued goal is to support the IVF community during COVID-19 pandemic and provide educational opportunities while bringing our community together. I personally have really enjoyed seeing so many reproductive biologists reaching out from across the world to participate in past sessions, as well as today, by logging in, listening, and asking questions. I am Dara Berger, and I will be moderating this session for you. We have an amazing program planned for the next 70 minutes. Depending on the volume of questions we receive from our audience and feedback from clinics adapting to the changing circumstances, we may continue this conversation in future sessions. Our theme today is SARS-CoV-2 and IVF Revisited. I encourage you all to ask questions using the Q&A section of the conference service. Please do not use the chat function for questions you want speakers to answer. This situation has become one that none of us could have probably imagined. As a result, many of you, including myself, have questions regarding how to run a laboratory that may be unexpectedly shut down and what to do when it is time to reopen. We therefore put together a very special episode of the International IVF Initiative. Today we will be focusing on attendee questions. International IVF Initiative has a talented team who is working behind the scenes to post your questions to our speakers. I encourage you to ask your questions, again, using the Q&A section of the conference service. Some questions will be addressed live, and some will be typed in the Q&A section directly, so keep an eye out for those. Please do not use the chat function for questions you want the speakers to answer. I will now turn the program over to Jacques, who will begin our discussion while you, the audience, create additional topics for us to address today, live and on Zoom.
Well, thank you, Dara. Hello, everyone. I would like to introduce you to an excellent panel. It's a panel of seven specialists. They will answer questions that you have posed to us a few weeks ago and in the interim until now, and also our live entry now for your Q&A session. So you can still keep asking questions. We've got a lot of questions, very good questions. The panelists today have bios. They are on our site. Possible disclosures in these bios are listed, so you can go and check that out. So the panelists are Alan Parker from Cardiff, Wales. Alan is a virologist. He comes to us via Giles Palmer. He knows him from being a well-known virologist in the UK, and he's known as the virus tinkerer. And thank you, Alan, for joining us. We have Alison Campbell, from the Care Network, lab director, runs about 12 clinics, has sometimes free time, and she's coming to us from Northern England. We have Lodo Parmegiani. Lodo, you remember from 2 weeks ago. He's, of course, an infection specialist as well as an embryologist and lab director. He comes to us from Bologna, Italy. We have again Kay Elder, also from Cambridge, also from the UK, Cambridge, and she introduced virology to us 2 weeks ago and did a great job. We have Kim Pomeroy from an undisclosed location in his RV far behind the mountains in northern Arizona. We have Pavlo, Mazur, virologist and embryologist. So we actually have three virologists here from Kiev in the Ukraine. And then we have Dean Morbeck, who runs about 5 clinics, I think, both in New Zealand. He's in Auckland, New Zealand, and he also runs clinics in Malaysia. So thank you for joining us. We have divided all the questions in about 5 themes. We'll go and start with themes that are close to us. These are themes that have to do basically with the laboratory and the questions that all of you have asked about laboratory issues and COVID-19. So the first team is called COVID-19 and the IVF lab. And so this is through all panelists. You're always welcome to raise your hands, panelists, so that it's a bit like the US elections, you know, where you see this all the time. So you can do that, or I just point out to you who should answer, who's most likely to answer the question. So the first one is, what to do if a patient is in cycle and tested COVID-2019 positive, or if a patient ends up being positive, what protocols should be put in place with their samples? If we can start with Ali, if you could lead the way here.
Oh, hi, good evening. Here in the UK, it's 8 p.m. So, yeah, great question. We have to stop treatments if we find we have a patient who's infected with COVID-19. We'd ask patients to let us know immediately. And here in the UK, we've ceased all treatments, but prior to ceasing treatments, we were communicating closely with patients to let us know if they had any symptoms and screening them on entry, taking their temperatures, asking them epidemiological questions. So yeah, we cannot proceed with treatment. We need to think about the patient's safety, staff safety, and of course, public safety as well.
Lodo, have you come across patients who are positive? And what would you do? Your microphone, you might.
Okay, so the only thing happened, of course, in Italy, we discovered to have this pandemic when we were working, and the only thing happened is that we discovered that a patient that she's pregnant now, she is with the virus. So when she did the treatment, we scanned there, we checked for the temperature, and she wasn't infected. But now we are informed that after the transfer, she's infected. So we will monitor this patient. And yes, we need to stop. We need to stop treatment. This is what we did in Italy. We stopped.
So in the United States, it's probably a bit more complicated and very much up to the individual clinics, or at least to some extent. Kim, what would you do if you have an infected patient who's running, who's about to have an egg collection?
I think it's very difficult to tell whether a patient's been infected or whether they are infected. We don't have great tests. We don't know false positive, false negative rates on these. And so it'd be very difficult. Using temperature by itself is not adequate either. So I think if you are forced for whatever reason, or you've elected for whatever reason to go through cycles with people that potentially could be infected, then the one thing I would do additionally, besides trying to safeguard the patient and the staff members through PPEs, is I would make sure that I drew blood from that patient and kept it as a backup, so that later when there are tests that are more accurate, or that we know at least how those tests behave, I'll be able to check it and find out.
Okay, thank you. Next question is, for labs that are open or that will reopen, what safety measures should be taken to protect lab staff as well as samples? So both the staff in the lab and the samples. And the extra question here is, please be specific and give us details. Dean, could you lead the way here?
Sure. So we're at a unique position here in New Zealand in that we've been in a lockdown. So it's a government-initiated lockdown that required us to stop. We're not an essential service. We're getting ready now to start back up. Hopefully everything's looking positive in terms of being able to go to what we call our alert level 3, which allows most services to reopen, but then to have in place social distancing. So that's where, for staff safety, we still will run with 2 shifts, as well as with PPE, masks, eye covers, and gloves that are changed frequently when engaging in areas that are at risk. And of course, then a complete clean down in between the shifts of all surfaces. There's some air exchange differences in between cases that are being done. So at least the guidance that's out there is at least 2 air exchanges complete, but I've seen as many as 7. And so that's how we're protecting both the patients and the staff.
Yep.
And then as far as gametes, we're currently not taking any special approaches to the gametes and embryos and the processing at this point, just because of the lack of evidence there that it's of a risk.
Yeah. So I hear goggles or glasses, I guess.
Yes.
Yeah, that's a good idea. Are these, they all have the same goggles now or glasses or?
Well, we're in the process of still procuring that, which is the challenge. Yes.
Next question. With the amount of washing that we perform during the laboratory IVF process, is the theoretical COVID-19 viral load in the transfer media a realistic possible risk to the patient? Okay, what do you think? You're on mute. Yeah, thank you.
Well, Kim raised his hand first.
Okay, yeah, Kim, why don't you go ahead?
I just wanted to say something really briefly, and I'll turn it over to Kay. And that is that David Battaglia did an estimation of the dilution that we have when we're going through culture, when we're going through also vitrification and warming. And that was dependent upon transferring 1 microliter with the embryos or the eggs into a 25 microliter drop for each of the procedures that we did. And his final dilution, he calculated, was 1 to 6.6 trillion.
Yeah.
So just in what we do, there is a high dilution. Now I'll turn it back to Kay to.
Yeah, so your argument is if you start with 10,000 particles, very unlikely to start off with, or 100,000, you're still ending up with a dilution factor in the billions, right? So yeah, okay.
Yeah, the other problem is we don't actually know what an infective dose is, as far as I know, at least.
Yeah, that's right. Yes.
You know, we know that it's transmitted by droplets. There's a suggestion that it may be transmitted by aerosol. But as far as what an infective load is, I'm not aware that there's much information. Perhaps Alan knows a bit better.
Alan, do you have anything to add?
I think Kay actually hit the nail on the head there for me. I mean, we know a limited amount about the infectious doses required to cause infection in by aerosol route, but in terms of other routes, I mean, this is going to be a recurring theme in this discussion. We just don't know yet. You know, these other routes, we don't know whether we'll be able to even infect cells, let alone gametes or anything else yet via that route.
Ali and Kim. Ali.
Yeah, I was thinking if we're talking about lab techniques and processing, assuming that there could be viral particles in the seminal plasma, don't we know that centrifugation can cause aerosolization? So I guess this, if this is all true, we need to be extremely careful when we're opening the tube that's been centrifuged and ensure it's always within a class 2 hood.
Right. Kim, you like to add? Or?
Yes. There's a very famous researcher named Bielansky from Canada. He has done a lot of work on trying to see whether infected embryos can pass on disease to recipients, for example. And he's done a lot of embryo transfers. He's done them with embryos that have been exposed in utero to viruses. He's experimentally exposed embryos to viruses, as has a number of other researchers, and in no case yet have they been able to produce infected offspring. So there's several things that have to happen for this to occur. There has to be enough virus there. The virus then has to be able to, when we're talking about SARS-CoV-2, it has to be able to bind the ACE2 receptor. It has to be able to have a furin enzyme there that will cleave the spike and then allow it to penetrate. And all of these things have to occur. And one of the big things that's probably lacking is lack of a receptor on the zona pellucida.
Yeah, yeah.
Yeah, can I just add to that?
Okay.
And again, I'll defer to Alan, but we don't have any reason to suspect that there will be virus particles in seminal plasma. I think it would be unusual for a respiratory virus to find its way into seminal plasma, but what does Alan think?
Yeah, I mean, Kay, again, I think you've hit the nail on the head there. I think it would be extremely unusual, and I don't know of any other examples where we've seen respiratory viruses existing in those other kind of bodily fluids. So I think it's highly unlikely. And as Kim has pointed out, to the best of my knowledge, although there needs to be further studies, no one yet has shown that ACE2 or any of the other putative receptors that SARS-CoV-2 can use or may be able to use are expressed on any of these gamete cells. So I think there's some very fundamental studies that can be done to put these things to bed and to alleviate some worries. And a little bit of basic science could go a long way here. A few weeks in the lab could show whether these gametes, these cells, express these particular receptors. And as Kim has also pointed out, I don't know much about trying to infect gametes or embryos, but it sounds like it's been attempted and it hasn't happened yet. So I think we probably don't need to worry too much about this possibility at the moment. I guess what we don't know yet, because we're so early on in terms of this pandemic, is if there are going to be any effects of contracting this on fetal formation during, when the mother is infected in the first trimester. And again, I'm going to come back to that saying that we just don't know enough about this virus yet, and time will tell. Obviously, we've seen babies born since people have been infected with it, and we haven't seen anything obviously in the later stages of gestation, but the early stages is too early to say yet.
No. Kim, you raised the points that you saw. You sent us actually a list of references. We have put those on supplemental material in this session, so people can go on the supplemental material and see the reference list. Those are actually, that particular part of the reference list are other references from Kay. Those have PDFs. That reference list doesn't have PDFs yet, but we'll work on that. So, I mean, the next question, you actually covered the next question, Kim, which is, have there been experiments on embryos and interaction with viruses being performed? And the only thing I looked up because of other questions that came up is the Van Roos article. I think that's part of your reference list. In 2000, biology reproduction was done in the bovine, scanning electron microscopy and confocal. And they looked at sizes of viral particles and imitated that with beads on the zona pellucida of the bovine, and found that the smallest pore size in the bovine it's pretty small, at about 180, 150 nanometers, and goes up to 223. And you would actually calculate from the small virus that they used the digestive tract virus. I won't repeat the name here, but it is 40 to 50 nanometers in diameter. So it should theoretically go to the zona, and it didn't. It was stuck in zonae, but it never went into the perivitellin space, or they couldn't find it. The corona size diameter is 120 to 160 nanometers. So that's a bigger virus than the one that they used in that paper. Kim.
Yeah, a good reference is Stringfellow's reference, or also my reference on the risk of cross-contamination in liquid nitrogen by Pomeroy. But when you look at what viruses have been studied, most of these have been in the bovine system, and they've looked at bovine retrovirus, they've looked at BVDV, which is a flavivirus, which is much smaller, as you mentioned, but is very similar to coronavirus in that it is an RNA positive stranded virus. And all of these have envelopes. So there are some similarities, I think, that we can get from looking at the bovine information.
Yeah. Okay, the next question. What would be a practical and economic protocol for utilizing UV in the lab to help kill the virus without harming any gametes and embryos? Dean.
I don't believe that there's much evidence for the use of UV.
Exactly.
Obviously, it can inactivate DNA. We use it all the time for that purpose. But we have really effective decontaminants such as ethanol and say for other products that have shown to be very effective against removing and inactivating this virus within a minute, depending on how it's used.
Lodo, you use UV.
Yes. Well, what I would say is there is a lot of confusion about UV. So, of course, when we give UV radiation, we need to give the adequate amount of UV dose to decontaminate the microorganism. Okay, so this means that, for example, we should have a lamp that is dedicated to the worktop, and in this case, as we do for liquid nitrogen, for example, we can be sure of the energy that we are giving, and of course we need to calculate this energy. If we consider virus with virus such as hepatitis, but also coronavirus, we need 10,000 UV dose, which is micron watt per second on centimeter square, which is something like 10 times lower the energy we need to deactivate a spore, for example, or the most resistant bacterium. So we have the technology, but we cannot say just, okay, we open the UV lamp and we are safe. So we need to calculate the UV that we are giving on the worktop. So this means that we should set up the procedure, and maybe people know that it's dangerous to stay in the laboratory or in the room when you are using UV. So that's the reason why it is not so easy. And then of course with UV, you are deactivating, you are breaking the DNA. Of course, you are not cleaning the virus, but this is enough. This is enough because, yeah.
Great, thanks, Lodo. Pavlo, a question for you. What's in your opinion the filters action that can be used in the lab? You think filters can eliminate
filters?
Yeah.
Well,
activated carbon.
Yeah, I think that that's only filters that can help, because activated carbon actually can absorb viral particles. But anyhow, the transmission of this virus is likely to be the contact ways. So we need to clean surfaces first of all, and well, the air is not well. If we wear all of us will wear masks and well, keep the distance, everything should be fine, I guess.
Okay.
I had a small addition about the virus and zona pellucida and attachment to all lemma. Well, it's just a small one, because well, if we'll try to intentionally transform human oocytes, we will fail, because there were several experiments with different type of viruses, and all of them are very inefficient. So at this point, at least for this RNA virus, we could be, well, calm, I guess.
Okay. Well, thank you. Dara, over to you for theme 2, which is restarting IVF cycles. Important topic that people are asking a lot of questions about. So, Dara, your turn.
Thank you. I would like to start by mentioning a document that is available to all IVF lab staff. I'm going to share my screen. The College of Reproductive Biologists covered feedback and suggestions from its members regarding IVF laboratory management. 2 checklists were released in the document titled IVF Laboratory Management During COVID-19 Pandemic. One checklist gives detailed instructions for laboratories that have stopped performing procedures. The other checklist provides information for laboratories who are performing IVF procedures. This document can be found on the AAB website as well as on ivfmeeting.com under supplemental material. Programs are encouraged to interpret the suggestions provided as best for their individual situations. These documents were created from laboratory directors, embryologists, and andrologists in the United States. If there are guidelines from other countries, please reach out and let us know. So that brings us to our first question for the panel. And it is, how long should labs wait before reopening? Should labs be open now? Should we continue to be doing emergency medical egg freezing? Allie, would you like to address that?
Yeah, well, it's a very difficult question, and there's so many variables and so many considerations. Personally, I think there need to be a number of labs open for emergency cryopreservation, both for sperm and eggs for oncology patients. I think that's very important. And in terms of when could we reopen? Well, again, that's going to be dependent very much on the nation who's making those decisions. But I think the first step towards that might be reduced restriction for travelling, and also the definition of essential medical treatment. So I would say as soon as travelling is permitted, then clinics could start to introduce very cautiously, and they need to adapt and be very flexible, but some at least diagnostic treatments, and go in a very cautious and progressive way.
Okay, thanks. Do you think we should test all patients for virus before restarting the IVF cycles?
To me, I think that might be necessary. I think if we've got a reliable test, it depends if you're talking about PCR testing for infection or immunity testing for antibodies. We know we don't have immunity testing that's fully validated yet. So either or ideally a combination of both to test staff and patients would be, I guess, the ideal, but whether that's going to be efficient or effective wholly, we don't know at this stage.
I have Kim and Pavlo who both want to answer as well, so I'll go to Kim first.
I really wanted to defer to Lodo because he addressed this very well and made it very personal about his own testing. So Lodo, could you weigh in on this?
Yes. This is what we are investigating now in Italy, because it's very, so we have the possibility to perform different tests, okay, for antibodies and for PCR, but at the very end only the PCR is recognized from health ministry, okay. So now we need to understand, to restart, if we would like to restart, which test we have to perform and when, because I think also this is very important, when we will test the patient, at the beginning of the stimulation or before they do the oocyte retrieval. So for example, in my case, I was sick with COVID and I did I tested the antibodies, but it was too early, and I was negative. And then at the end of the virus, I was tested with PCR and I was negative.
Dean, what do you think?
Well, I think we could also look towards transfusion medicine as a lead on this, because they deal with this in a much more sensitive manner, because they're taking blood donations, and donors are really at low risk, like our patients are at low risk. We do the screening for them beforehand, and the blood donation centers currently are not testing. But if someone turns out to be positive after they've donated, at least in Italy, I've seen there's just a recommendation where they will discard all of the blood products from that, but that there is no additional testing done, and partly because that's a major load on the testing system. And I know we wouldn't allow, like in New Zealand, they wouldn't allow us just to start saying we're going to test patients based on our own wishes, because they have their own triage system for who gets tested.
So that's interesting that you mentioned that, because the next question is, should from now on patients have a positive serology for SARS-CoV-2 antibodies, so only patients vaccinated or those who have had the virus be accepted for treatment?
I would just, if I'm still on board here, I'd say no, that's not realistic for us to be able to do that. And I think where the biggest risk is when you look right now at New York City, when it's really chronic for that area, then we have to know that it's so prevalent in the community, and we have much higher concern because it affects the healthcare system. Whereas the rest of the world is having to figure out, how do we kind of do a dance with the fact that it's out there and it's at a lower level, and we're not going to be able to test everyone. We don't have good enough testing. We can't possibly afford to do all of that. So we have this dance, and it's going to be going back and forth between, you know, how protective we are, but we can't possibly stop patients without having good testing for it.
Mm-hmm. Kim?
Yeah, I would say, why would you test everyone as a prerequisite? We don't test everyone for all diseases, and we don't even know if this is an STD, which it most likely isn't. So I think there's a lot of questions we have. We're not going to know the answers to them because we don't know exactly how the virus behaves. We don't know when its antibodies are elicited in humans, IgG or IgA. We don't even know I don't think we know the time scale even of when you can detect the actual RNA. So there's a lot of questions we can't answer until we have this information.
Okay, fair enough. So then the next question, if we're moving on, we're going to start treating patients. Does anybody have any suggestions on who to treat first and how to go about recontacting patients whose cycles may have been previously stopped?
Pavlo was waving his hands around.
Yeah, but it was the previous question, not this one. Okay, just small addition. When we test patients for presence of RNA of virus or antibodies, there is no guarantee that there will be no reinfection of the same patient with another strain of the same virus. So we cannot know. On the other hand, what should we do with the patient with the flu, right? So we don't test everyone for antibodies for flu virus. Right, so it's just the same. The patient should be healthy, right, to go into the cycle, and that's all we need from this patient.
Ali, did you have comments about how to contact patients going forward?
Yeah, I was thinking the best way to say which patients would you treat first, assuming that everybody's ready and there's a big bolus of patients waiting for treatment, then the fair thing to do would be to treat those patients that you'd already had to cancel, that were already in the system, prioritize those patients, and also those ones with who would have a negative effect from a further delay. So potentially patients of advanced maternal age, those patients that could be really compromised by any further delays.
Okay, thanks. And social distancing for patients. Does anybody have any suggestions for that? How do we start seeing patients, have them in our offices, and what recommendations could be made to clinics about social distancing for patients?
Well, I can answer that, Dara, because we're addressing that currently as we prepare to get started again. And we also are doing this in Malaysia with our clinics, which haven't shut down, and that is to limit how many patients are in the clinic at one time, so that the distancing is definitely maintained, that patients are wearing masks, PPE, primarily masks, and then that we also limit access to partners so that we don't need extra people there. So that means that in our case, it's the patient, the woman bringing in the semen sample. We're not having the couple come together.
Okay, thanks. One quick housekeeping item before I hand this back over to Jacques. Some of our audience members has been asking why not all the questions are being answered, but they are being transferred over to the answered section of the Q&A portion. We are working on answering all the questions, and if you don't see your answer pop up there or be discussed here, we will follow up after. All right, over to you, Jacques.
Thank you, Dara. Well, in the interest of time, very short questions on the third theme, which is COVID-19 cryopreservation and cryostorage. 5 questions, and I would prefer short answers. Should we keep recently frozen samples in a quarantine tank and until we confirm patients are not COVID-19? Kim, what do you think?
I think that's a very conservative thing you can do. If you have those facilities, go ahead and do it. But I don't think it's necessary.
Loda, what do you think? Loda, you're muted.
Sorry. I don't think it's sorry. I don't think it's necessary because I know. I think most of all probably know what is my way of thinking. We should be a procedure to make sure that what we are freezing is doing in a safe manner. And then if the problem is the virus in the around the oocytes or the embryo, I think that we don't know, but the risk is very low. So, but if the problem is the contamination of the tank and the cross contamination, or just we need to wait. I don't think we need to do it. Yes, we just need to wait until we are not safe to do the embryo transfer. This is what we have to do. Wait.
Right. Well, I think looking at the questions that I had here in this section, Dara, I think most of those have been answered. So I'm going back to you on asking the questions regarding this theme on cryo and cryo storage.
So I am actually going to go to a new document and ask a question. Sorry, it just moved. Would it be prudent to periodically test waste or debris from our liquid nitrogen storage dewars for a battery of the most concerning pathogens? Kim?
Can I answer? Okay, Kim. No.
Oh, go ahead, Lodo. I know you know this well too.
No, okay, well, this is for our two, yes. I don't think so. I think we need to demonstrate that because for sure we will have contaminants in our cryo tank, but the titles of this contaminant won't be enough to create problem, potentially. But what we have to do is try to demonstrate that we set up procedures and to demonstrate that we are cleaning periodically. And as we said also in the webinar, the solution is probably the dilution of the risk, so dilution of washing of the samples at warming, okay? Because otherwise it's impossible. We can test the treatise in our tank, and then after a few weeks or few months, we may have another contamination. So we have to consider that potentially the cryo tank have contaminants. I don't know if Kim is think the same thing. I don't know.
Yeah, I agree with you. To me, it would be like testing people to see if they have MRSA in their nose. We know that most people have MRSA, flesh-eating bacteria in their nose. But just because it's present doesn't mean it's going to give you, flesh-eating bacteria.
Okay, thank you. The next question is, should we consider all patients as infected if there is no possibility to test them? So we need to store the samples, sperm or oocytes and embryos in separate tanks.
So, okay, I can. The idea is that we potentially have to consider all our patient infectious, because maybe we are worried about COVID-19, but we may have other potential contamination. So what we have to do to set up our procedure, if we choose to use for example, for vitrification open carrier, maybe we should be sure that at least the cryo storage is made in a safe manner with a double bagging or with a carrier that is closed for cryo storage, and demonstrate also that we are working in a clean environment. And of course, I can disclose my conflict of interest, but I strongly suggest to sterilize the liquid nitrogen for any handling and any vitrification and warming.
Okay. Jacques, are you can I throw this back over to you for a couple questions?
Sure. I have a question here from Colin Howells. On a country basis, what needs to be done in order to quickly open up again services to help people have a family again? Who are the decision makers? So is that the government? Is that the local government? Is it the clinic, the university? Who makes the decisions here? 5 countries here, Ali, yeah.
Well, in the UK, the final decision is made by our regulator. So at the moment, we've been issued with a directive to stop all treatments. And we would potentially lose our license if we didn't adhere to that directive. The HFEA have been advised by our societies, the British Fertility Society and the Association of Reproductive Clinical Scientists. And I'm pleased to say that at least it feels very collaborative. It feels in the UK that the clinics, the societies and the regulator are working really carefully together, and that we will end up eventually with a risk-based strategy for resumption of treatment.
So how is this in New Zealand, Dean?
Yeah, it's clearly directed by the government. So, you know, we're currently in a 4-week lockdown where we're not an essential service. We were able to stop cycles or to complete cycles that were already started. We will be able to start up once we go to alert level 3, which again, I hope is in about 10 days. One interesting caveat to that is if I travel anywhere starting after that time, I go into a mandatory 2-week quarantine in a hotel.
Right. Lodo, how is it in Italy? I think I know the answer, but go ahead.
Yeah, we need to wait. I agree with Dean. Our procedures are not urgent, and then we need to wait until the regular medical activity, surgery activity restart, and then we can restart. But of course, we need to wait. We need to wait the government. Today we had a communication from the Prime Minister say that the lockdown will continue until the 3rd of May.
3rd of May. Okay. What about this country?
Well, everyone in this country is doing it like in the Wild West, and they're deciding themselves whether they're going to shut down, when they're going to open up, and what criteria that they will use. In fact, I think most people in the US know this, but maybe other people don't. It became very divisive here in the United States. 2 major groups, ASRM and another group that formed, that felt differently about whether they should shut down or whether they should not shut down. And so in the US, basically people are left to do what they think is best.
Yeah, and there are different levels of authority, of course, right? It's not only the professional organizations, but you have the CDC, you have the FDA. They haven't said much, but the New York State Health Department, which is always quite opinionated, just yesterday came out with a new directive that basically states, and it's a little bit hard to read and open for interpretation, but it basically states that fertility services, including infertility treatment and procedures, are considered essential services. That's since yesterday. So that's quite different from what the ASRM, I think, has advised us. And then they say the additional resources are the ASRM document. So it's really interesting. It's incredibly different from, so we get different messages.
Yeah, you know, Jacques, one thing that I don't think we've discussed very much, I don't think we need to discuss it, but people need to be aware, we might start using a lot more volatile chemicals now in our laboratories. I hope that we don't. But
Yeah.
for example, in the past, I would never use ethanol in my laboratory.
Yeah, exactly.
And so we have to think about that. And also people that have shut down have to think about what's built up in their laboratory, how they're going to re-sanitize it, how they're going to go ahead and get it running again and give it enough time so that any of the volatiles can be moved out of the lab that happened during the shutdown.
Yeah, and ethanol goes right into the culture medium. It partitions right into it. Lots culture medium and goes through the oil. So yeah. Okay.
A question or two if you'd like to pass it back to me.
Okay, go ahead. Yes.
Okay.
Passing it back.
With so many citizens not working or furloughed around the world, do you expect to have a delay in patients returning as they may not have the funds or insurance for a couple of months, or do you think it's going to be the opposite?
I think I'll start. We're expecting now, of course, we have a unique situation in New Zealand as well, in that we have some public funding, so they get to start without that, you know, risk of not having the funds, even if they're not employed. But clearly, just like after the recession of 2008, there was an impact on how many people could afford or were willing to take the risk of putting the money into that. So I think it will have an effect on number of patients.
Kim?
Yeah, I think what we're going to see is an initial big rebound in the United States, because a lot of people have already paid for their cycles, have paid for their treatment. And then as soon as it opens up, a lot of people are going to be waiting in line and go through that. And then afterwards, I think we're going to see a decrease in the number of patients that we'll be serving.
Lodo, did you have something you wanted to add?
No, I would say that our doctors say that they don't have a lot of requests in this moment. So they are doing, you know, a visit by web visits, but they say that they don't have a lot of patients. But maybe this is a situation only in Italy. My colleagues in Spain said that they have a lot of patients that are waiting to be treated, and they also think, you know, that Spain have is a good resources also for donors, and I also think that in the future they will have more donors. Unfortunately, due to the situation, and maybe they are thinking that more young girls will be available for donation for the economic crisis.
So jumping around to another question from online, what about the possibility of injection of the virus into the oocyte during ICSI? Is there any information on whether the virus, if injected into the oocyte, could integrate into the oocyte's genome and get activated? Pavlo?
I think we should answer to this question together with Alan, but well, we should have like enormous loadout of virus to have any reaction from the oocytes. Besides, it's RNA virus, so it cannot integrate into the oocyte genome. Still, we don't know the possibility of this particular virus to reproduce inside the oocyte, but well, we don't have any clues that some of the same viruses did the same. So you can possibly inject it, but you need to have an oocyte surrounded by the only lane in the medium consisted only from the virus to get in this oocyte. So because there were different experiments with adenoviruses, and they tried to infect the oocyte with a huge loadout of adenovirus. It's DNA virus. So, and they failed. Only this tiny bit of oocytes got this virus. So when you're talking about this coronavirus, it's very unlikely. And Alan, please.
Yeah, if I can jump in there. So I think Pavlo has said much of what needs to be said there. This is an RNA virus. It hasn't been reported to integrate. I think it's extremely unlikely it will integrate. Of course, I can't really, unless you were doing, obviously, a highly controlled scientific experiment, why would you ever want to microinject these things into an oocyte? The other thing to say, of course, is, you know, it will be extremely non-physiological to do so. So you would gain essentially a hypothetical risk if you injected a huge dose of this inside the oocyte as to what integration might happen, if any. But of course, you would have to offsetting that against the fact that the virus, probably in its natural form, couldn't infect the oocyte anyway. So you could come up with a hypothetical risk. As Pavlo said, similar studies have happened with adenovirus, which is a DNA virus, and perhaps is more likely to integrate, and that has come up with an extremely low rate of incorporation into the genome. But I think that will be several orders of magnitude higher than we're likely to see with an RNA virus such as SARS-CoV-2.
Thank you. So in terms of egg banking, donor sperm, donor eggs, is there anything additional we should be testing donors for, since now there is a new virus on the scene? Kim?
I assume you were looking at me, Dara.
You know, I wasn't, but this is your question.
Okay, yeah. Working with an egg bank, I can't see why there would be anything at this point that we would require an addition. I think we would maybe keep those specimens isolated at least until we know a little bit more about the virus. But even then, I think that's not a very probable event that this virus is going to affect eggs. So I can't see much. The one thing I mentioned before is I would make sure you had serum on all of your donors so that you could go back to that serum later and see whether they had potentially been infected.
Dean, did you have something to add?
Yeah, just I think, you know, if we follow again the blood banks, maybe as a donor facility, being able to, if someone shows up positive in the next few days after a collection, then you are faced with that decision of what to do with the donated tissue. And again, since we don't know, even the blood banks, that, you know, they need to start providing results for testing of the blood that came from those individuals to say, was it present? Was there viremia? Because we expect viremia to occur once there's a significant load of virus already, which usually is after onset of symptoms. So I think, you know, the testing is going to have to be agile around those kind of conditions. We take healthy people in, we screen them with questionnaires, and then we may have to adapt depending on what happens after that.
Okay, thanks. Jacques, do you have some questions prepared?
Yeah, a question by Alex Woodyard. How can antibody screening be utilized to allow treatments to resume? I mean, complicated. And, you know, I'm not sure that has anything to do with resuming. It may have to do with staffing, maybe. What do you guys think? Dean, you were shaking your head.
Well, I think it would be great once we get to that point. And it's really going to be, you know, what are the epidemiologists and what are the public health systems doing in general in terms of screening for this as it evolves? And we don't have an answer for that one.
A question by Alison Bartolucci. We do require staff to have a flu vaccine. Using that philosophy, and in the absence of a vaccine, should staff be tested? And then the question really should be, well, should it be tested? How often do you test? You know, once a day, once a week? You know. Any comments? Any answers here? Suggestions?
I'll again say I think we have limits on how much testing we can do and how effective it is. You know, the problem is we're going to be using PPE throughout the clinic.
Yeah.
We'll wear masks all the time. Yeah. I know, you know, Mayo Clinic's requiring cloth masks for those not interacting with patients. There's always going to be that in the interim.
Yeah. Lodo, you had something.
No, I agree with Dean. I agree with Dean. So,
okay. So would there be a situation here, one question, would there be a situation where you would just freeze all embryos to play it safe and wait later for testing? What do you guys think about just doing that instead of also fresh embryo transfer? Ali?
Yeah, it depends where we are in this process of this journey of recovery, I think. I think freeze all is a very safe and successful. I think whilst we've got a burden on the main hospitals, it makes some sense to freeze embryos rather than to do a fresh transfer, primarily because of the risk of OHSS, albeit small. We don't want hospitalizations from that syndrome. So I'd say it depends really very much on where we are in the process.
A question here by Abdeli Zokmar. I hope I'm pronouncing that right. When you have one member of your team, like an embryologist or a lab anthologist, who is positive or maybe even has symptoms of COVID-19, what do you do? Right? I mean, we're not exposing ourselves just to other colleagues. We're also exposing ourselves to others outside the clinic. So this thing can happen. So what do you do, Dean?
Yeah, so we're still really trying to sort out how this will look, you know, whether you stand down an entire team. And part of that will be revolving around what the government is doing in terms of contact tracing and how they define what happens if somebody's within a close, what we call bubbles here. Then that whole bubble will be affected until the you know, the 14 days can be isolated. But I think, you know, what we're doing as well is saying how much can we keep staff from not interacting much, and we're hoping that that's going to be enough, along with PPE, not having a break room where people can sit together, practicing all the good practices that we now are doing, you know, routinely. That should make it so we don't have to stand down a whole team.
Yeah,
but we don't have that answer.
Yeah. Well, in the case of you having several teams, which is, I think, what most people will do, is to have a longer day. Are you going to adjust those days? Are you going to say to the clinicians, you can only do one egg retrieval every other period that it takes you to do an egg retrieval? How are you going to take care of this? How are you going to, Kim?
Yeah, of course, it always works well to go to the physician and tell them, no, we're not going to do that work. That's not going to happen. It's never happened in the past, in the US at least. I think you're going to find out that people are going to have staggered groups, and they're doing it now in the US, where half of your team will work or a third of your team will work for a week or 2 weeks, because we're definitely going to have more people quarantined than we're going to detect that are going to be infected.
Yeah.
And once they're quarantined, anyone that's been in contact is probably going to be quarantined. So that's how you plan for it today, at least. In the future, I don't know.
So what happens if this is a sudden event and now suddenly everybody in your lab is being quarantined from this moment to the next? 1 minute later, the news comes through, tests, family members, everybody's going to be quarantined. Now you have the incubators full with embryos. What are you going to do? I mean, really?
Yeah, no, I'm going to go there and I'm going to freeze them. I'm going to have one person go in there and freeze everything.
Okay.
I'm just like if a hurricane was coming and I was in Miami. Okay.
I was just worried if it was all your teams that are affected and not just one team. But a lab a lot of labs are very small and only have one team, one embryologist. So tricky.
Yeah. I mean, yeah. I'll just concur with Kim. You know, there will be an extensive, drastic measure that you might have to take. Even with two teams, it's conceivable that both teams suddenly are affected, and if they're affected to a degree that you have to stand them down, then you just have to do like an emergency preparedness measure of freeze-alls. Yeah.
Okay. Anybody would make a phone call to the nearest clinic and help out, ask them to help out? Is that a possibility?
Yeah, I think so. I think during this period, it's unbelievable how cooperative and collaborative everybody's been. We're all in the same situation, and we all want to help each other out. So absolutely, I think that's exactly what would happen.
Yeah, and particularly in towns and cities where there are at least 2 clinics, right? You would expect that kind of collaboration is possible. Okay, only another 100. While we were talking, only 100 questions came through, but they're kind of repetitive. And I wonder, Dara, if we should go to teams 4 and 5. We are now 1 hour and 1 minute. Are you guys up to it, panelists? Fresh? Yeah, okay. We'll go to team 4. Is that okay, Dara? Yeah,
yeah, that's fine. I'm going to squeeze in one more.
Okay, go ahead. Yeah. You saw one, yeah.
What are the known risks for the fetus in the first trimester? Or during pregnancy at all? Does anybody have any?
Alan, I think, right?
Yeah, well, I think we've kind of touched on this already. You know, the virus was only first described, I think, the 31st of December last year. So there haven't been any pregnancies yet that have come out from where the person may have been infected from the first trimester. So until we get through the next 2 to 3 months, we aren't going to know. I think, you know, I don't want to put my head on the chopping block here, but I think it's unlikely it's going to be the kind of virus that's going to integrate or cause any issues like that. But we just don't know. We only know that obviously people have contracted this virus during the later stages of pregnancy, and those babies appear to be, there's no correlation with any sort of malformations or anything. But obviously the first trimester is all critical, and we won't know that for another 2 or 3 months at least.
Yep.
Hey, Alan, would we know anything based on what happened with the original SARS-CoV-1?
Well, the original SARS-CoV-1 burned itself out pretty quickly because it was so lethal. So we don't, as far as I'm aware, know, or at least I've not read anything about malformations as a result of people who were pregnant and went through that. So to the best of my knowledge, there's no correlation with the original SARS-1 virus, but the data there is very small because it was very lethal, that one, and very few people were pregnant who contracted the virus. So it's a difficult one. This one is obviously a lot less lethal, which means it can spread more readily, which means we are going to get more data on this. We are going to see people who are infected during that first trimester, but we don't know yet. We'll know in a few months' time.
Okay, so the first question under theme 4 is, does COVID-19 have a specific host cell? What are the first cytokines activated in the immune response against COVID-19? Alan?
So yeah, I mean, I think this is pretty well documented already. It's a respiratory virus. It infects via ACE2 primarily, which is expressed abundantly on lung epithelial cells. So that is the primary target. The virus is inhaled. It infects the cells at the back of the throat initially, and then obviously moves down into the lungs. And we are seeing increasing cases of people with cardiovascular issues related to COVID. And that's because ACE2 is expressed at high levels in the heart. So that's the natural tropism of this virus. In terms of other potential routes of infection, you will probably have to look at where else ACE2 is expressed. It is expressed in a few other places. But obviously the virus being respiratory is going to hit those cells via the respiratory route that are expressing ACE2 primarily. And so that is largely the lung epithelial cells. I guess if it sheds through the lung and can affect the heart, then it will do too.
Do you think it will have, oh, Kay.
You mentioned cytokines. There's a recent comment came out in Nature Reviews just yesterday or today. It was entitled, how does COVID-19 kill people? And it mentions that the cytokine pathway that's involved involves interleukin-6. And interleukin-6 is primarily trying to recruit other members of the immune system. And so at the moment, there are some trials of interleukin-6 antagonists underway. But I think I sent that paper to Thomas. It's just briefly mentioned, it was either yesterday or today.
Yeah, so I missed that part of the question, Kay. You're absolutely spot on again there. IL-6 is one of the first major cytokines that's expressed, like with many other viruses, and I do think there may be a significant benefit to anti-IL-6 therapies potentially. So it'd be interesting to see how those trials pan out.
Do you think there are going to be any long-term effects on fertility? Pavlo?
My favorite answer to this question, we don't know, but well, we don't know for now. But we can see, we can look over the other coronaviruses and other respiratory viruses, and well, at least we know some information from them, and probably we will not need to wait for this unpleasant event. But well, we'll see. I guess we'll see it soon. But for the other coronaviruses or flu viruses, orthomyxoviruses, paramyxoviruses, well, there is no such thing as these unpleasant events.
Okay, I think that the only thing that we can say now is that this COVID-19 will have an effect on epidemiologic fertility, because we won't have kids in the next for the next 9 months, because we stopped to work. Yeah, this is what we can say now.
Thank you for that point. Jacques, do you have a few last questions to get in?
Yeah. Since most of the IVF consumables and media are internationally shipped, should we be concerned about contaminated services like packaged materials, etc., also inside the boxes, inside the sleeves? I mean, we would assume that the processes in those manufacturing places will be completely changed. We can, of course, inquire, but anybody, any thoughts here? Any worries? Lodo?
Yeah, sure. It's always a problem of procedures. So the manufacturers, I think you received an email from manufacturers that's some weeks ago saying that they were trying to move the manufacturing in zone with low contagion. Now all the world is exposed. So what we have to do is to have procedures to when we open the boxes and inside they cannot be contaminated.
Yeah. So we expect them to explain their new procedures, right? Yeah.
Yeah.
Jacques, can I just say?
Kim, go ahead, Kay.
Can I say that any material that's coming from the outside, from any country or even within the same country, is potentially contaminated with all kinds of microbes, not just this particular virus. So every lab should have in place a very strict protocol before allowing any external material to come into the lab. Yeah.
Kim.
Yeah, I was just going to say things like plasticware, a lot of the hard things that we get that are discardable, those things are usually gamma irradiated. So those I would not worry about. I'd only worry about the outside packaging and how you handle that. And it may be something as simple as you have a hood that you're not using that has a UV lamp in it, and you leave these things in the UV lamp for a while. That may be something that may work, but you'd have to, test it.
Oh, sorry, Dean, go ahead.
Oh, I'm sorry, Jacques. I'm a little bit skeptical of using the UV light on the plasticware because that will change the properties of it and release VOCs from UV of plasticware. So I wouldn't recommend that, at least what you're going to use with gametes and embryos. But I do wonder, and I don't have an answer for this, I'll pose it out to the rest, is we know that the amount of virus that one is exposed to is important. And so how much is going to be on a contact material that's been shipped? And how likely will that be to actually be infectable to someone? I don't have the answer to that, but I have to imagine it's not very, because we do know that the worst cases that we see for individuals, healthy people, is when they're exposed to high doses of the virus. And if you're exposed to very little, or just a small amount that's on a surface, is it really, we have a lot of fear right now, but without a lot of knowledge about what is the real risk.
So when plastic and carton and paper, when they're exposed, metal, when it's exposed on purpose, the number of hours it takes for the virus to, still be active, it's always in, I think, 3 days.
3 to 9, but is it, while being active, is it really readily infectable, infective?
Yeah, so those are experiments where they use enormous numbers of particles, of course, to make it easier. And that really is probably not reflective at all of getting a package from a culture medium manufacturer that's being sent to you. But you change the rules or change them for the better. Still labs, you see boxes in labs, carton boxes in labs. See it all the time. That needs to probably change. They need to keep us out of the labs and unpack there and then handle it going inside. It's all a matter of space too, and space is essentially precious. So I understand there's difficulties there, but we have to adapt all these protocols. Okay, we're 10 minutes over 4 here EST. Dara, I give it back to you. Thank you, panelists. Give it back to you, Dara, for wrapping it up. Thank you very much.
Thank you very much. I was lucky to work with a great team of embryologists and lab directors in the community to put together the IVF laboratory management during COVID-19 pandemic document that was discussed today. I want to thank them, along with the panelists who answered some really tough questions. Uncharted territory is challenging, and I'm glad to be surrounded by such experts. I also want to thank the audience whose participation today made this session one of our best. Happy Passover and Easter to those of you who are celebrating. And Jacques will tell you about our 5th session that is coming up next.
Yeah, our 5th session is on Tuesday, this coming Tuesday, that's April 14. It's at 3 o'clock EST, so same times wherever you are. So Dean, it's another early morning for you and late evening for you, Pavlo. This session will be on in vitro growth IVG of human ovarian follicles. It's a very exciting topic, and they're going to talk about future developments. It's hosted by Professor Richard Anderson, and the speakers are David Albertini, Dr. David Albertini, and Professor Evelyn Telfer. So that's the coming Tuesday. It's a session organized by Colin Howells. So please visit ivfmeeting.com website and let us know if you have any questions also about this topic. It's very likely that in 3-4 weeks we come back to this topic. People will have more questions. We'll have more answers too, as most of you have indicated. There are a lot of things we don't know, but we'll get some answers in the next weeks or months. So we'll come back in 3 or 4 weeks, could be 5 weeks. Remember, we're here to support you, but support us too. Spread the word. Again, cheers to our panel, Ali, Dean, Alan, Pavlo, Kay, and Lodo. And of course, thank you, Dara, for being a co-moderator here. And thank you, audience, for your incredible questions and for joining us today. See you soon and stay safe.